Let's take a minute and look at the differences between metaphase of mitosis and metaphase 1 of meiosis. Remember, every cell that goes into the cell cycle goes through this process called S, which is DNA replication. During DNA replication, the entire DNA molecule is copied. And during that process, the DNA is in the chromatin form. But for ease of discussion, we're going to look at images where the DNA is in chromosome form, just to make it easier to visualize. So we're going to start off with a parent cell, and we're going to make this parent cell as simplistic an organism as possible. We're going to say that this eukaryotic parent cell has only one type of chromosome. It's a diploid organism, though. So for that one type of chromosome, it has two copies. It has one that came from the father, paternal, and one that came from the mother, maternal. So each one of these chromosomes has the exact same DNA in it. It's just that one version was inherited from the father and one version was inherited by the mother. They are both the same type of chromosome. Here's where we're going to introduce some new vocabulary. Anytime you have two of the same type of chromosome, you can call that a homologous pair. Homologous just means the same. The DNA in this chromosome is the same DNA as this chromosome. It's just that the alleles might be a little different. They have all the same genes. They just might have different versions of the genes. The father may have had slightly different alleles for each gene than the mother had. And now this parent cell has inherited one copy from father, one copy from mother. Same type of chromosome, just two copies, a homologous pair. Now we're going to send this parent cell into the process of cell division, into the cell cycle. It's going to go through G1, it's going to go through S, and its DNA is going to get replicated. That means each one of these individual chromosomes is going to get copied. And that's going to produce this. It's hard because a lot of students look at this X-shaped drawing and think this is a chromosome. This is actually a replicated chromosome. This is also a replicated chromosome. We've already gone through S here. So in the parent cell, after it has gone through G1 and S and G2, as it comes into M, this is what it would look like inside the nucleus. All of the DNA has been replicated. You could call this right here a replicated chromosome. Over here, a replicated chromosome. We refer to this copy as a chromatid and this copy as a chromatid. So these would also be called sister chromatids. These two copies are identical. Over here we also have sister chromatids. These two copies are identical. When you take this replicated chromosome, these sister chromatids, and compare them to this replicated chromosome, these sister chromatids, it's the same type of chromosome. So this and this are also called a homologous pair. It would be better to call these a homologous pair of replicated chromosomes, but very few people do that. Now, why did I just go through all of that? Well, remember, during metaphase 1, we don't see chromosomes lining up head to tail. We see them lining up side by side. So this is what the parent cell would look like during metaphase 1 of meiosis 1. The homologous pairs of replicated chromosomes are lined up side by side. This is a pair that maybe came from mother. One came from mother and then got replicated during S. One came from father and got replicated during S. Same type of chromosome, same genes on these, just different versions of genes. This is a homologous pair, and they're going to line up side by side during metaphase 1. Over here, you can see the chromosomes look different. This is a longer one, right? Different DNA on these chromosomes. Some was inherited from father, some was inherited from mother. These have been replicated. This is a homologous pair of replicated chromosomes, and they're going to line up side by side. Compare that to metaphase of meiosis. In the metaphase of meiosis, we just line up head to tail. This slide is difficult to see, I know. There's a lot of information on it, but I included it in here so that you can do a side-by-side comparison of mitosis on the left and meiosis on the right. There are two divisions in meiosis. This is the meiosis 1 in the light yellow, and this is meiosis 2 in the dark yellow. The artist has drawn the mitosis side by side with meiosis 2 because these look identical. It's just that on the meiosis side, because we've already gone through meiosis 1 and then a cytokinesis, we have two daughter cells here, and they're each going to go through meiosis 2. But what's happening during mitosis looks just like what's happening in meiosis 2. It's meiosis 1 that's very different. It's meiosis 1 where crossing over is going to occur during late prophase 1, and where homologous pairs of chromosomes are going to line up during metaphase 1. Crossing over is going to cause bits and pieces of DNA to switch and swap between the maternally inherited chromosomes and the paternally inherited chromosomes. So what used to be all red and all blue is going to end up a mix of red and blue. Remember, you are genetically different from your parents. You are not a copy of your mother, and you are not a copy of your father. You are not exactly half of your mother and exactly half of your father. Crossing over is a very randomized process. You got a mix and match of bits and pieces of each parent. That's why you are also not an identical genetic copy to your brothers and sisters, unless you are an identical twin. So, late prophase 1, that's when crossing over occurs. Metaphase 1, that's when homologous pairs of chromosomes line up. During anaphase 1, we're going to separate those homologous pairs. During telophase 1, we're going to make two new nuclei. And then we're going to have a cytokinesis. And now we've produced two daughter cells, but we're not done yet. Because remember, meiosis needs to create haploid cells. These daughters are still diploid. They still have two copies of each type of chromosome. So now they're going to go through another metaphase where the chromosomes will line up head to tail, another anaphase where the chromosomes will be separated out, another telophase where new nuclear envelopes will be created, another cytokinesis where now we have daughter cells that only have one copy of each type of chromosome. They are haploid. Both mitosis and meiosis are followed by this cytokinesis. Again, cyto translates as cell, kinesis translates as movement. When early scientists were watching cell division under the microscope, they noticed that at the end of mitosis and at the end of each division of meiosis, the cytoplasm started to move and a line of division appeared. This line, this contractile ring that forms around the center of a cell during cytokinesis, this is actually made out of microfilaments. It's microfilaments that line up underneath the membrane and then contract. They get shorter and shorter and shorter. And this contractile ring tightens into the cell. It's like tightening a belt around a cell. Eventually, the belt will get so tight that these two halves will split apart from each other, creating daughter cells. Now, the other term you should be familiar with is cleavage furrow. This little ditch that gets created where the contractile ring is referred to as a furrow, a cleavage furrow. One other thing to note about cytokinesis, what it looks like in an animal cell is not what it looks like in a plant cell. Plants have cell walls. So instead of dividing up the cytoplasm in such a very obvious way during cytokinesis, what plant cells do is simply build a new cell wall in the middle of this parent cell and divide it in half that way. On this slide, we're looking at a cell that is going through the process of cell division. It has entered the cell cycle. It has gone through G1 and S and G2, and it's now gone into M, nuclear division. This cell happens to be an example of the stem cells that are going to produce gametes. So this cell will use meiosis for its M. It's actually two parent cells, one stem cell that's going to produce sperm, one that's going to produce eggs, or oocytes as they're technically called. Remember, something unusual happens in the female during the production of the eggs. Let's start over here on the male side. This is a parent cell. It's a stem cell, and its job is to go through cell division and produce sperm. The sperm have to be haploid. They have to have all of the individual types of chromosomes, but just one copy, because they're going to come together with eggs that all have one copy of each type of chromosome. When the egg and the sperm come together, now we will have an embryo that has two copies of each type of chromosome. So in this parent cell, this happens to be an organism that has two types of chromosomes. We're going to call it a long chromosome and a short chromosome. This is a diploid organism, so it has two copies of each type. One that it got from mother, that's in red, and one that it got from father, that's in blue. This organism inherited one long chromosome from its mother, one long chromosome from its father, one short chromosome from its mother, one short chromosome from its father. Two types of chromosomes, two copies of each. Now, when it goes through the first meiotic division, when it gets to late prophase 1, these chromosomes, which will be replicated during the S phase, that's why we see them as X's now instead of individual lines. They're going to cross over during late prophase 1. You can't see that on this slide, but really what we would be looking at in metaphase is a mix and match of red and blue. So somewhere in here, this cell went through early prophase 1 and then late prophase 1. The reds and the blues, after they've been replicated, are going to line up and cross over, and we're going to get a mix and match of red and blue. Now, during metaphase 1, homologous pairs are going to line up side by side. This is a homologous pair of replicated long chromosomes. This is a homologous pair of replicated short chromosomes. They line up side by side, and then they get separated. They get pulled apart. So everything on this side is going to go to this end. Everything on this side is going to go to this end. New nuclear envelopes will be made. A cytokinesis will occur and divide this cell in half. And by the end of meiosis 1, we'll have this, two daughter cells. Not gametes yet, though, because they still have two copies of each type of chromosome. And gametes need one copy of each type of chromosome. So now these two daughter cells are going to go through meiosis 2. They're going to go through prophase 2. They're going to go through metaphase 2. They're going to go through telophase 2, and then cytokinesis. And this is going to be the result. 4 individual sperm cells, 2 from each daughter. And inside each sperm cell, there is one of each type of chromosome. Now, the artist has drawn them as red and blue, but remember, they would be a mix of red and blue. Every single individual gamete is unique. It has its own unique set of versions or alleles of genes. That's why you not only are not an identical copy of your parents or of your siblings, but you also will never bump into a genetic identical copy of yourself anywhere else in the world. You were produced from the combination of two gametes, a sperm and an egg, and those gametes themselves were unique in terms of the combination of alleles that were inside of them. Now, let's take a look at the unusual situation in the female. Here's the parent cell. Here's that stem cell inside the ovary. Don't worry about these technical terms, by the way. I'm not going to test you on those. Here's that stem cell in the ovary. It's going to go through meiosis 1, and it's going to produce two daughters, just like what we saw in the male. But something strange is going to happen. One of these daughters is going to essentially survive. The other daughter is going to degrade. It's going to shrink down and die and become what we call a polar body. Under the microscope, polar bodies get very, very small. They are essentially just the chromosomes wrapped in a piece of membrane. There's no other material in here by the time they're formed, and they are not viable. So this thing cannot divide anymore. But this is a viable cell. So this will go on into meiosis 2 and create again one viable daughter and one non-viable daughter, another polar body. So by the end of meiosis, this female parent stem cell is not going to create 4 eggs like we saw in the male, but 1 egg and polar bodies. And these polar bodies are going to just be degraded. This is one of the reasons that eggs are so very precious from an evolutionary standpoint. Females make many fewer eggs than males make sperm, and females make eggs through only a very limited time when they are in their mother's womb. Remember, the fetus, the female fetus, makes all of the eggs that female person is ever going to have. This all takes place during fetal development in the female. The male is very different. The male will begin producing sperm when he hits puberty, and he will continue manufacturing new sperm throughout the rest of his life. We're going to switch gears now and go back and talk about some of the unique features of each stage of the cell cycle. First, I want to introduce some new vocabulary. The time it takes for a cell to get through one round of the cell cycle gets a name. We call that generation time. So the time it takes for a parent cell to go through the cell cycle, go through cytokinesis, and create 2 daughter cells, that's referred to as 1 generation, and that's generation time. Now, the generation time varies among different types of cells, and that probably makes sense to you. There are some cells in the human body, for example, that divide very rapidly. Think about, for example, your skin cells. Your skin cells replenish themselves constantly because older cells at the very top surface of your skin are constantly being sloughed off as you live your life every day. The lining of your intestine, the lining of your respiratory tract is also constantly being replenished. There are also certain blood cells that are produced very rapidly in the human body. The parent cells or the stem cells that generate sperm in the male also divide quite rapidly. Now, by rapidly in a human being, we're talking about a generation time of about 8 to 12 hours. There are also cells in the human body that can be thought of as dividing very, very slowly. There are cells that only divide once every few months, and even once every few years. Very, very slowly dividing cells. For example, the cells of the liver in an adult human being are very slow to divide, about once a year. What's interesting to note, though, is that these slowly dividing cells can divide more quickly under certain circumstances. In other words, they can be stimulated to divide quite rapidly. Now that tells us something about cell division. That tells us that cell division is regulated. There are ways to speed it up and to slow it down. A good example of this is in the human liver. The human liver, like several of our organisms, is quite redundant. In other words, you have more liver than you actually need to survive. You can live with about a third of your liver present. So when a person undergoes a liver injury. For example, maybe getting into a car accident and having their liver broken or lacerated. Up to 2/3 of the human liver can be removed, and the person can still survive. And over time, what's going to happen is that 1/3 of a liver is going to grow back into the size it was before the accident. Remember, liver cells don't normally divide very often, but the injury process is going to trigger a change in those cells, and they're going to start to divide quite rapidly until the liver is replenished or replaced to what it was. When we look at cells from different parts of the body and we compare their normal generation time, one of the things that we notice is that most of the variation occurs in the gaps. In other words, a very rapidly dividing cell next to a very slowly dividing cell shows most of the difference in the gap stages. This makes sense if you think about it. If you have two cells that both come out of a human body, and one of them is dividing rapidly and one of them is dividing quite slowly, it's going to take the same amount of time in both of those cells to copy the DNA molecule. There's only so fast that DNA can be copied. So the S phase of the cell cycle doesn't tend to change in length from cell type to cell type. Similarly, the M phase tends to be a relatively set length of time from cell type to cell type. Where we see the differences are in the two gaps, and it is especially gap 1 that varies in length among different types of cells. Now, there are some cells in the human body, remember, where once they're in place in a mature adult, they never divide again. Neurons do not divide. Muscle fibers do not divide. So what happens in those cells is that from G1, they simply go into G0. They simply arrest. They stop dividing. There are other cells that divide so rapidly that the G1 becomes essentially shorter than you can imagine, virtually eliminating it. So it depends on the cell how long the generation time is going to be. Most of the variation in generation times comes in the G1. In rapidly dividing cells, G1 is very short. In slowly dividing cells, G1 is very long. In cells that don't divide at all, G1 simply moves into G0. There's very little variation in the S phase from cell type to cell type, even when generation times vary a lot. And there's very little variation in the M phase as well. Now, we've learned quite a bit about the cell cycle and its different stages by examining cells not as they exist in the actual organism, but by taking them out and growing them up in the laboratory in cell culture. We know, for example, that during the gap stages, everything in the cell, with the exception of the DNA, has to be copied. That means all of the cytoplasm and its contents, the cytosol, the actual fluid, all of the organelles in the cytoplasm, all of the proteins and other molecules that are dissolved in the cytoplasm, all of it has to be replicated during G1 and G2. Now, there is one important exception that I want us to be familiar with, and that is in the embryo. Remember, for a human being, an embryo is created when a sperm cell and an egg cell come together and fertilization occurs. The one-celled embryo that is created is called a zygote. And that embryo then will begin to divide. The zygote, the one-celled organism, will divide into two cells, and then that will become a four-celled embryo, and then an eight-celled embryo, and on and on and on through many, many divisions until all of the cells are in place, and now we just have an immature version of a human that we call a fetus. And then the fetus continues to grow and develop until it's time for birth. Now, when we're looking at just that fertilized egg, that one-celled embryo, that zygote, what makes it really unique is that it contains enough cytoplasm, enough dissolved molecules, enough of everything, in fact, for many, many cell divisions to occur. So in the egg, in the embryo, I should say, the fertilized egg, each round of division through the first several rounds creates smaller and smaller and smaller cells. That original one-celled zygote has enough cytoplasm so that we don't need to spend time replicating cytoplasm and dissolved chemicals. We can get through divisions very rapidly without spending a lot of time in G1 and G2. We've already done all that replicating. And all of the replicated materials are present in the egg before it gets fertilized. This is unique. Most of the time when a cell divides, through each round of the cell cycle, the cytoplasm and its contents have to be replicated. The only cell that does this differently is the egg. The egg already has enough cytoplasm and enough materials in the cytoplasm so that that amount of replication during the cell cycle doesn't have to happen. On this slide, you're looking at some pictures of the first divisions of a human embryo. You're also looking at actual images under the microscope of this process. So again, over here on the left, this is a fertilized egg, or what we call a one-celled embryo, or also called a zygote. This is the most primitive form of a brand new human being. Now, what will happen within about 24 hours is the first division. It will go through the cell cycle. It will go through cytokinesis, and you'll have two cells inside of this shell structure that exists in a human. Now, within another 24 hours or so, each one of these cells is going to go through the cell cycle and it's going to divide. Now you have 4 cells in there. And then after that, you're going to have 8 cells in there, and so on and so on and so on. Notice that from this cell to these cells, these have gotten much smaller. And then these, when they divide, they're going to get smaller. And then when they divide, it's going to get smaller. In other words, with each round of cell division, no new cytoplasm is being made. The cytoplasm that was present here was already plenty for all of these cells.